Oh yeah, that would be great.
Okay, you're on.
I'm trying to let.
I'm gonna wait. I'll wait till it starts. I won't start the recording yet. Waiting for Katie.
Yeah.
Hi, Gary.
Hey there. Hi, George.
Yeah, we're all on here. Good to see everybody.
Okay. Well, good to see you guys. We're ready for the show to start.
Oh yeah.
On the committee for just a few years now, and she's done a tremendous job in her work on MS-related OPCs that you'll be hearing about. And we'll soon be starting a postdoc over in the School of Public Health looking at human retinoids, not retinoids, brainoids. Retina's in my brain, unfortunately, and looking at COVID virus, coronavirus, as I understand. And we're all looking forward to your presentation. So go for it. Thanks very much.
Thank you.
And one other thing I think, as Itzy mentioned, if everybody could put their computers on mute so we can have 110% attention to her words. Thank you.
Thank you, Don. Well, hi everybody. Again, thank you for joining me in this exciting day for me. And a mi familia y amigos de México, gracias por estar presentes. Today, I will be presenting the results of my thesis project, and it is about how induced pluripotent stem cells from people with multiple sclerosis can differentiate into oligodendrocytes under nurturing conditions, but not under inflammatory states. Before we walk through my project, I want to give you a condensed, brief overview of it. And many of the, two of the many challenges that MS patients experience and the problems that we have as a field is that there is a failure to remyelinate, and there is a high variable outcome of neurodegeneration. And we don't know why some people remain stable throughout the disease while others quickly progress into disability. And to gain a better understanding of these high variable outcomes, we study the ability of induced pluripotent stem cells, iPS cells for short, their ability to differentiate into myelin-producing cells from MS patients with varying degrees of disability under homeostasis and also under inflammatory conditions. And I have divided my talk into 3 parts. In the first part, I will be presenting an overview of multiple sclerosis, the aspects that gave rise to my research and how we approach them, and the goals of this endeavor. For part 2, I will present the research, the results, and which includes the generation and characterization of oligodendrocytes under homeostasis and also under inflammatory conditions. Part 3, I will cover the highlights of my research. I will acknowledge the people who have contributed to this work, and we'll have a discussion at the end. MS is an autoimmune disease that affects the central nervous system, which consists of the brain, the optic nerve, and the spinal cord. It causes neurodegeneration via inflammation, axonal damage, and myelin loss. Myelin is a fatty insulating material that is produced by cells called oligodendrocytes, and it coats axons, and this facilitates the propagation of electrical impulses, which is the way information flows from one cell to the next. And in a system that is constantly computing incredible amounts of information, myelin has a crucial role in communication efficiency and therefore how we function. MS falls mainly into two main categories: relapsing remitting and secondary progressive. Relapsing remitting is the most common course of the disease and is characterized by attacks, the relapses, followed by periods of complete or partial recovery, the remission. And most people will progress into secondary progressive, and this type is characterized by a worsening of the neurological function over time. In MS, myelin is attacked by the immune system, causing inflammation, axonal damage, and demyelination, creating lesions known as plaques. And these areas are damaged areas where messages coming from the brain or the spinal cord or any part of the body are not passing through, or are passing very slowly or in an erratic way. And the implications of this communication disruption depends on the area of the central nervous system being damaged. And the extent of the impact in terms of neurological outcomes is highly variable. Some people may experience mild symptoms and a stable course, while others will experience severe progressive disability. It is thought that a small fraction of people with MS can remyelinate to some extent, but for most patients, this process is hindered. And remyelination tends to be more effective earlier in the disease than later. And this is where failure to remyelinate is a main contributor to disability. But the importance of myelin goes beyond conduction. It also provides metabolic support to neurons, and it protects the axonal integrity. We know that chronic demyelination can cause permanent axonal damage and cell death. Then because of this, there is a keen interest in developing therapies that would enhance the innate ability of the central nervous system to repair. But unfortunately, these efforts have not been successful in human trials. As I just mentioned, the central nervous system has the ability to repair. It has an available pool of oligodendrocyte progenitor cells that could differentiate into mature oligodendrocytes. And a single oligodendrocyte has the capacity to produce 40 to 50 myelin segments. And this process is a multi-step process that depends on the ability of the oligodendrocyte progenitor cells to migrate at the site of injury, to proliferate upon arrival, and to be able to interact with demyelinated axons in order to differentiate. And this process is also influenced by factors such as cell signaling, debris clearance, and the disease environment, which may affect any stage of this process. And while we know that all of these factors are at play, we still don't know the mechanisms behind successful and failed remyelination, and whether there are innate differences in the ability of these cells to repair is also unknown. It has been more than 100 years since multiple sclerosis was characterized, and we still don't know the reasons behind the highly variable outcomes that I have mentioned. So in order to gain a better understanding, we hypothesized that failed remyelination could be reflective of inherent defects in the generation and or the maturation of oligodendrocyte progenitor cells and oligos in people with multiple sclerosis. And if this is the case, how can we approach this question? We approach this question through a stem cell technology, which allows to generate oligodendrocytes directly from MS patients as they live through the disease, and also allow us to bypass restrictions from post-mortem studies and also interspecies differences that may arise from animal models. So here's a question. If we take stem cells from MS patients with varying degrees of disability, would those varying degrees of disability be reflected in the ability of those stem cells to differentiate to oligodendrocytes? And so we defined 3 groups: healthy controls, stable multiple sclerosis, and progressive multiple sclerosis. And then we took peripheral blood mononuclear cells, or PBMCs, and those were genetically reprogrammed to iPS cells, to induced pluripotent stem cells. Then we took those stem cells and direct their development towards oligodendrocytes. And after 3 intensive months, we get oligodendrocytes directly from MS patients with varying degrees of disability. And now we can study and see what differences, if any, there are between and across the groups. And the goals of this project was to investigate the basic biology of the cells, pathologic characteristics, including developmental, genetic, and functional differences, and of course, compare all of these findings across the groups. As you now understand, my project depended in the ability of the iPS cells, the induced pluripotent stem cells, to differentiate to mature oligodendrocytes. And as such, we needed to make sure that we were initiating with the correct, right type of cell. And so after the PBMCs were reprogrammed to iPS cells, the cells went through quality control screening to validate the new cellular identity and also ensure genomic stability. So fully reprogrammed cells gained expression of TRA160, which is a marker of stem cell marker, and they also showed normal karyotypes. The timeline to go from iPS cells to mature oligodendrocytes is 3 intensive months. And during this process, the cells express specific markers that depend on the specific stages during development. So in each developmental stage, the cell will express specific markers that allow us to monitor their transition to maturation and their successful differentiation. And what you see, as I will show you in subsequent slides, we routinely screen for OLIC2, PDGF, receptor alpha, O4, and MVP. And what you see in these images are the morphological changes that the cells go through this process. And as labor intensive as this was actually a rewarding experience for me to see how the cells change almost by the day. And they start as a monolayer of iPS cells colonies that quickly expand, and they start piling up to form sort of 3D structures. And they start forming 3D structures. And very early in their development, we dissociate this. We mechanically dissociate this layer to induce a sphere formation and allow the cells to keep their differentiation process while the spheres grow and while they are in suspension. And at day 30, we plate those spheres and allow them to adhere to the wells. And this adhesion triggers signals to the cells and direct them to emigrate from the spheres. And this sort of cellular exodus happens in waves. Usually neural progenitor cells, neurons, and astrocytes come out first. Okay, then in order, as I mentioned before, the cells go through the developmental stages, and they have specific markers that we can use to track and monitor their development. And in order to make sure that our cells were reaching those developmental milestones, we use these specific markers for the stages. And so for the oligodendrocyte progenitor stage, we screened for PDGF receptor alpha and for OLIC2. For immature oligodendrocytes, we checked O4 and OLIC2. and for mature oligodendrocytes, MVP, PLP, O4, and OLIC2. Also, from the genetic standpoint, we use single-cell RNA sequencing to corroborate the identity of the oligodendrocyte progenitor cells. And we sorted the cells at day 60 based on the expression of PDGF receptor alpha. And through single-cell RNA sequencing, we obtain individual profile expression of the cells, and then through gene clustering analysis, identified patterns of gene expression. And what we found is that more than 92% of the cells were positive for PDGF receptor alpha, and which this corroborates that the cells were oligodendrocyte progenitors. The next developmental milestone that we checked, of course, was that the cells were reaching maturity, that they were becoming mature oligodendrocytes. And at this point, we checked for canonical markers such as OLIC2 and MVP, but we also assessed for morphology. And as you can see, the cells were able to obtain both. They express proper markers and morphology. And also, we were also able to sort cells at the immature oligo stage by O4, just a marker specific that I just mentioned. And after about 3 weeks, we immunostained these cells to identify O4, and we were able to confirm that the cells were able to keep the expression of this marker through maturity. And we know, and there is a specific, there's a hallmark of oligodendrocytes that is their ability to wrap around axons. So we also screen for functionality, because it is not enough for the cells to produce myelin. It is crucial that they are able to interact with axons and to unsheath them. And to this end, in order to assess myelination, we co-culture oligodendrocytes with neurons, and then identified by immunostaining MVP, which is myelin basic, myelin basic protein that you see in red, in green, and this identifies mature oligodendrocytes. And in red, you see neurofilament, which is marker to identify axons and neurons. And as you can see, the cells were able to interact with the axons and apparently unsheath them. But to unambiguously show this hallmark of myelin wrapping around axons, we evaluated our cultures by electron microscopy. And you can see in these images that the cells wrap around the axons. And what this is telling us is that the cells coming from MS patients were able to differentiate to oligodendrocytes, and also were able to myelinate. Of course, we also validated and characterized specific markers by flow cytometry, such as O4, PLP, and OLIC2. And we also get genetic confirmation of MVP, of myelin basic protein, by qPCR. And for the next set of experiments, we focused in O4 and OLIC2 as our identifier markers. O4 has been used to characterize oligolineage cells directly from tissue of healthy individuals, and also from chronic lesions from MS patients. And OLIC2 is the transcription factor that is necessary to activate the genetic program of oligos. We were also very interested in conducting a more robust quantitative analysis of this differentiation process of stem cells to oligodendrocytes. And we achieved this through flow cytometry by screening for O4 and OLIC2 of large populations of cells. And we found that, as you can see in this image, that all of the cells were able to differentiate to oligodendrocytes to different, with different efficiencies. And some cell lines were, as you could see, show greater variability in their differentiation process. However, when we combined the donors per group, and we averaged the experimental replicas per donor, it was evident that there was no differences across groups in the ability of the cells to differentiate and express O4. We also obtained similar results from OLIC2. And at this point, we could tell that the cells from MS patients were able to differentiate to oligodendrocytes, and that the disease group made no difference in this ability. The cells also were able to express specific markers, and they developed proper form and function. And so based on these experiments, we were not able to conclude that there were innate differences in the ability of the cells from MS patients to differentiate to oligodendrocytes. Okay, this is great. All the cells were able to differentiate to mature oligodendrocytes, but under nurturing conditions. As we know, multiple sclerosis is chronic and it's also inflammatory. It generates a hostile environment for long periods of time, years really. And this is a very important consideration for myelin repair. And in order for us to mimic a chronic and inflammatory disease environment, we expose the cells to interferon gamma, which is a cytokine that has a major role in multiple sclerosis because it has a direct effect, a direct impact in the immune attack against myelin. And so if by subjecting the cells to sustained exposure to low dose gamma, we can study the ability of the cells to differentiate under this inflammatory state. And by the way, low dose was defined as 10 nanograms per ml. So we established parallel cultures of untreated and treated cells. Some cells were treated from day 36 to day 85, other cells from day 60 to day 85, and some others from day 36 to 60. Most of the experiments you will see are cells that are at the end of culture. That means that they are mature oligodendrocytes and they were collected and processed day 85 approximately, but they were either treated at day 36 or at day 60. And what we found is that sustained exposure to low dose gamma had a profound inhibitory effect on the ability of the cells to differentiate to oligodendrocytes. And again, the group didn't make a difference in this ability. And as you can see, day 36, the effect is particularly strong when cells were treated at day 36. And interestingly, if we waited for the cells to mature a little more and get to the oligodendrocyte progenitor stage, and then treated them with interferon gamma, the effects were milder, and a portion of the cells were able to recover and differentiate. And this is really interesting because what we're seeing here and what this data is telling us is that there is a period during development where the cells are highly vulnerable to the effects of interferon gamma. And they cannot cope with an inflammatory effect. We also found similar effects for OLIC2 and corroborated the detrimental effects by genetic analysis by qPCR, and we see that gamma effect is equally bad for both MS groups. And also, again, what this is telling us is that constant inflammation is really profoundly inhibitory for the cells, and it's telling us that it doesn't matter that the cells have the ability to differentiate to mature oligodendrocytes and the capability to remyelinate if an inflammatory environment is blocking both of these processes. As we were doing experiments, we noticed that despite the fact that the cells were not becoming oligodendrocytes, we were not losing cells either. The total cell count was pretty much the same under all conditions. And we thought this was unusual because we know that interferon gamma can inhibit differentiation by causing cell death. So we did a count of live cells, and we found that low dose gamma didn't have a global toxic effect on the cells. Doesn't matter the group or the conditions. And also this lack of toxicity has been shown in animal models when under low dose gamma, that is. At this point, we have learned that interferon gamma has a profound inhibitory effect in the ability of the cells to differentiate. So the cells are not becoming oligodendrocytes, yet they're not dying. But we still don't know, we still didn't know what was happening with the portion of the cells that were not becoming oligodendrocytes. So we also know that several groups have shown in rodent studies that interferon gamma can inhibit differentiation by keeping the cells in the oligodendrocyte progenitor stage. So at the end of the cultures, of course, we screened for PDGF receptor alpha, and we found that, as expected, as the cells reach maturity, PDGF receptor alpha decreases the expression. And we also checked for astrocytes because we know that they can be a common occurrence in cultures and they can overgrow the culture. So we checked for astrocytes and also found that just like O4 and MVP and OLIC2, astrocytes are inhibited by interferon gamma in a very similar manner. And of course, at this point, you may have anticipated that we also check for neuronal markers, specifically beta-3 tubulin, also known as TOUCH1. And again, very interesting results, not because it's my research, but really interesting, that we found a concomitant increased expression of this neuronal marker, TOUCH1, with a decreased expression of O4. Again, the effect is particularly noticeable if cells are treated at day 36. And it seems that, at least in a portion of the treated cells, interferon gamma is redirecting the lineage phenotype to a more neuron-like cell. Excuse me. As we were studying the long-term effects of the inflammatory environment created by gamma, we also wondered about the short-term effects of gamma. We wanted to study the early transcriptional effects to be able to understand in what other ways gamma may be altering the genetic signature of oligodendrocyte progenitor cells. And for this set of experiments, we sorted OPCs at day 60 from three samples from progressive, 2 from stable, and 2 healthy controls. And then we allowed the cells to recover and then treated them with low dose gamma, but only for 16 hours. And we found that short-term gamma had a profound effect in the transcriptome of the oligodendrocyte progenitor cells. And 3,200 genes were differentially expressed. Among the highly upregulated genes, interferon gamma promoted genes involved in antigen processing and cross-presenting signaling. And this is very interesting because it suggests that OPCs might be acquiring immuno-like phenotype, that they may be acquiring characteristics of antigen-presenting cells. And the impact of this, the significance of this data is that it suggests the possibility that OPCs may, at the site of injury, might be converting to an immuno-like cell that may be stimulating and propagating damaging response while converting away from a cell that was designed to repair. And so globally, our research shows that short-term and long-term inflammatory environments have a profound detrimental effect in the abilities of these cells to differentiate and be able to repair. So the highlights of the research. So we have seen and we learned that iPS cells from MS patients with varying degrees of disability were able to differentiate to mature oligodendrocytes. And this was possible in the absence of inflammation. Chronic exposure to interferon gamma severely inhibits the ability of these cells to differentiate to oligodendrocytes. And it seems that it redirects the lineage phenotype to a neuron type. And the short term greatly affected the transcriptome of the cells. And it seems that the cells may be acquiring, the possibility of the cells that acquiring an immuno-like characteristics. And finally, the clinical relevance of this research is that it suggests that chronic low-grade inflammation may profoundly affect the efficacy of regenerative therapies. And it is very important that these therapies are tested under these inflammatory environments. And finally, I would like to acknowledge the people who have contributed to this work. Obviously, mainly, I want to thank my thesis advisor, Katie Wardenby, for her guidance and her constant presence and helping with everything regarding my project, of course. Calabresi's lab, everybody who has helped me in many ways, not just specifically with my project, but I want to specifically thank Dr. Peter Calabresi and Matt Smith. He did several experiments of the RNA sequencing with me and the bioinformatics analysis. And Lapchan for the co-culture of neurons and oligodendrocytes. Kathleen Fitzgerald for the statistical analysis. My thesis committee. And thank you, all of you, Zach, Basso, and Peter Calabresi. Of course, my PhD program, CMM. Thank you, Leslie and Colin, and the funding I received in order to for this project to be done. And with that, I thank you, everybody, and I'll take some questions.
Yeah, I have a question. Actually, I have two questions.
Sure.
Is the incidence of MS higher in people with inflammatory disease, IBD, Crohn's, very heavy smokers?
Well, there are, yes, some environmental factors are involved, and yes, obesity, smoking are a couple of those environmental factors.
Okay. And then the other question is, should this really be considered one disease? Genetically, it's really quite diverse, and that presumably could have a big impact on where the phenotypes go, although, as you point out, they end up with this problem in oligos, possibly through somewhat different routes. Do you have a comment on that?
I don't know the specific answer to that, but I think it is more than one disease. I think as the pathology progresses, it becomes more than one disease.
Okay, thank you.
Sure.
Very nice talk.
Thank you.
Hi, Itzy, here's Lena. Very nice talk. I have a question about what do you think of, so you were looking into oligodendrocytes only, but MS is mainly also peripheral nervous system, which is affected as well. So what about Schwann cells? They have slightly different mechanism, how they perform myelination in peripheral nervous system. So whether you would expect the same effect of inflammation, and whether you address that question.
The specific answer to that, I don't know. I would imagine that would be similar, since most of the proteins and genes expressed by the Schwann cells are very similar, and one of the main differences is the peanut protein in the myelin from Schwann cells. But it will be interesting to study that I actually have read a couple of papers on Schwann cells and their ability, and it seems that they have a greater ability to repair, but I don't know in the context of these experiments.
Thank you.
Itzy, I have a question. Great job. By the way, Lena, I may be wrong, but I think that MS primarily affects the CNS. I think it has pretty minimal effect on the peripheral nervous system. But my question is, Itzy, your data is showing that the inflammation is promoting more of a neuronal phenotype. Wondering if, do you know anything more about what type of neurons or any more characterization of that neuronal phenotype?
I don't know. Actually, when we were performing more experiments on this and more analysis, but then COVID hit, and so the only characterization I did was for touch one.
Okay, great.
Yes. And I'm sorry, I apologize. I think I didn't hear Lena's question very well based on what you just answered, Don.
Yeah, I could be wrong.
Yeah, the question was whether they're also affected in the peripheral nervous system. The Schwann cells are also affected.
Oh, okay. Yes, I don't know.
And is there any stuff in the literature about other inflammatory mediators? Do they have a similar effect on differentiation and myelination?
For
Other inducers of inflammation?
other markers of inflammation?
No, other inducers, inflammatory.
Oh yeah,
molecules.
there are many cytokines that can cause inflammation. We've only focused on interferon gamma because it has been shown that when patients are having flares, the concentration of interferon gamma increases. And also, yeah, mainly we just focus in interferon gamma because of that reason, but there are many cytokines that are involved in the generation of this inflammatory hostile environment in MS. Yes.
Great. Thanks.
Don, it's Katie here to answer your question. I think Itzy had also started to do some work with TNF, and it looks as though in at least some patients that has a similar effect as well. But again, that wasn't in a state to present thanks to the shutdown. So
Great, thanks.
I guess. Well, if there are no more questions, thank you so much, everybody, for joining me in this very exciting day. Bye.
Congratulations, Itzy. Stay safe.
Thank you, Bob. Thank you so much.
Bye, Izzy. Great job.
Great job. Very good. Good luck with everything.
Congratulations.
Thanks.
Yeah. Enjoy the champagne.
Good job.
I will.
Thanks a lot. This has been great.
Thank you.
Thank you for allowing us to listen to you.
Absolutely. Thank you for joining.
George said something, but he had his mute on.